We can see more data on acute convalescent samples later during the session. And the next speaker is Dr. Dustin Glasner from University of California, Berkeley, and his talk is Dengue virus NS1 induced endothelial cell intrinsic vascular leak is independent of inflammatory cytokine but dependent on endothelial glycocalyx integrity from Dr. Harris' lab.
Good morning, everybody. Yeah, that original title was a bit of a mouthful, but we've tightened it up a bit. Thank you for being here. I spent some of the most formative years of my life 30 miles south of here doing research, so I'm really happy to be back here in Maryland, and I'm excited to talk to you today about some of our work. So, Dustin Glasner, I'm a fifth-year PhD candidate in the lab of Dr. Eva Harris at the University of California, Berkeley School of Public Health. And I'm here to talk to you a little bit about our attempts to elucidate the mechanisms that drive NS1-induced pathogenesis. So as we all know, dengue virus is the most prevalent mosquito-borne virus worldwide. Over half the world's population is at risk of infection. There are 4 related serotypes, and the virus is made up of 3 structural proteins and 7 non-structural proteins. We're particularly interested in non-structural protein 1. NS1 is the only protein that's secreted by virally infected cells, and it's secreted as this hexameric barrel-shaped protein. We know it plays a role in viral replication and immune evasion. It's been implicated in pathogenesis through activation of complement. But recently, we and others have found a direct role for NS1 in pathogenesis through induction of endothelial dysfunction and vascular leak, both in vitro and in vivo. Clinically, we know that higher levels of NS1 are correlated with more severe disease, and we think this is because of a potential direct role for NS1 in pathogenesis. So far, there have been 2 major pathways identified that contribute to NS1 pathogenesis. The first was described by Dr. Napif Modiran in Paul Young's lab, and this involves activation of TLR4 and induction of pro-inflammatory cytokine secretion from PBMCs. There's also a cytokine-independent pathway that we discovered in our lab that involves the endothelial glycocalyx layer and induction of endothelial intrinsic enzymes in endothelial cells. So the purpose of this study was to tease apart the relative contributions of each of these pathways to NS1 pathogenesis. So the first thing we did was establish a model of vascular leak, of local vascular leak in our lab, and we adapted the Miles assay, and what we do here is remove the hair from the dorsal dermis of mice. We can then do intradermal injections of controls such as PBS as a vehicle control, VEGF as a positive control, and different concentrations of NS1 as experimentals. We then follow this with an injection of a tracer. This is an example of the types of data we get out of this. So this is the back, the tissue of a mouse following injection with Evans blue dye, and you can see PBS induces very low levels of Evans blue extravasation, whereas VEGF and NS1 induce higher levels of leak. And we see the same thing with Alexa conjugated dextran. This is a quantification of this data, and again, you can see low levels of leak induced by PBS, higher levels with VEGF, and a dose-dependent induction of vascular leak with NS1. And you'll notice that NS1 from a related flavivirus, West Nile virus, does not induce vascular leak in the dorsal dermis of wild-type mice. And you'll also notice that the data with our Alexa tracer is a little bit tighter and gives us a little bit more sensitivity. So that's the tracer we used for our further experiments. So going back to these 2 different pathways, we first wanted to look at the role of cytokines in NS1 pathogenesis, and we started by looking at the ability of human dermal endothelial cells to produce cytokines when stimulated by NS1 in culture. And what we found is that LPS induces high levels of both IL-6 and IL-8 secretion from cells, but NS1 does not. You'll also notice that there's no secretion of TNF-alpha from these cells, and we think this is because most endothelial cells require a secondary inflammatory signal in order to get cytokine or TNF-alpha production. So we wanted to evaluate the functional importance of cytokines in this endothelial-specific model. So the assay we use is called transendothelial electrical resistance, or TEER. And in this assay, we culture endothelial cells on a transwall membrane. We add a tracer of choice, or a molecule of choice, in this case either soluble NS1, inflammatory cytokines, and we measure the electrical resistance between the basolateral chamber and the apical chamber, and this is a proxy measurement for endothelial permeability. So as endothelial permeability increases, TEER values decrease. So using this model, we found that NS1 alone is capable of inducing endothelial hyperpermeability in human dermal endothelial cells. The same can be said for IL-6 and TNF-alpha. And And we can block that cytokine-specific effect with a monoclonal that's specific to these cytokines. But then when we use the same monoclonal antibody in conjunction with dengue NS1, we don't see any reversal of the NS1-induced hyperpermeability in these cells, suggesting that at least IL-6 and TNF-alpha are not involved in this endothelial intrinsic mechanism of vascular leak, or endothelial permeability. So then we took this into our in vivo model, and we evaluated knockout lines of both TLR4 and TNF-alpha receptor. And here we have, we have quantification of the mean pixel intensity on the y-axis, and then we compared vascular leak induced in TLR4 and the TNF-alpha receptor knockout mice to our wild-type animals that you saw in our earlier slides. And what you'll see is that there really are no differences observed between any of the conditions with either knockout when compared to wild-type animals. But we still see the same pattern of higher levels of leak induced by VEGF and a dose-dependent induction of leak with NS1. So this suggested to us that TNF-alpha and TLR4 signaling are not required for vascular leak observed in our local dermal model. Taking this a step further, we have some preliminary data looking at systemic vascular leak. So what we do here is we inject mice with a bolus of NS1, and 3 days later we inject with Evans blue dye as a tracer, and we measure Evans blue extravasation in both the lung and the liver, which are two major organ sites of vascular leak. And when we look at the wild-type mice, you can see that there's higher levels of leak in the lung and liver induced by NS1 in wild-type mice. And when we look at the TLR4 knockout mice, there's really no striking difference between the vascular leak observed between our TLR4 knockouts or our wild types. Then going even a step further, we looked at an infection model, and in this model, we either infect a type 1 interferon receptor knockout mouse or a double knockout of type 1 interferon receptor and TLR4. And when we infect with either a lethal antibody-enhanced dose of virus or a lethal high dose of virus, again, we see no differences in either morbidity or mortality between these mice. Okay, so now going back to those two pathways. We've talked about the cytokine-independent dependent pathway a little bit, but now we're going to talk about the cytokine-independent pathway. And in 2016, we published a paper that identified sialidase, cathepsinel, and heparinase as important regulators of vascular leak or endothelial permeability driven by NS1. And we implicated the endothelial glycocalyx layer in this pathway. And the endothelial glycocalyx layer is a complex structure of membrane-bound proteoglycans that lines the luminal surface of blood vessels and protects blood vessels from shear forces. It helps mediate blood cell endothelial cell interactions. It's involved in inflammatory signaling and has been implicated in a variety of disease processes. And what we found was that NS1 addition to endothelial cells induces activation of sialidases, which leads to cleavage of sialic acid from the surface of endothelial cells. It also leads to upregulation of cathepsin L activity, leading to an increase in expression of heparinase, which will then cleave heparin sulfate proteoglycans from the surface of endothelial cells. In that same paper, we also showed that inhibiting these enzymes was sufficient to block that decrease in tear that we observe when we add NS1 to endothelial cells. So we did the same thing with dermal endothelial cells and see the same pattern. So this inhibitor cocktail of sialidase inhibitors, cathepsin L inhibitors, and heparinase inhibitors is sufficient to protect endothelial monolayers from dengue-induced hyperpermeability. So we used the same inhibitor cocktail, and we brought this into some of our confocal microscopy readouts. And we looked at sialic acid, cathepsin L, and heparin sulfate expression on the surface of cells. And what we find is that dengue NS1 induces a decrease in sialic acid expression on the surface of endothelial cells, an increase in the expression of cathepsin L activity. I know it's a bit hard to see, but it's all quantified over here. And then we also see a decrease in heparin sulfate expression on the surface of dermal endothelial cells. Now, when we treat with the same inhibitor cocktail that I showed you here, we see reversal of these effects. We see normal distribution of sialic acid and heparin sulfate and a return to basal levels of cathepsin L activity in the cells, suggesting to us that these enzymes are involved with some of the glycocalyx-specific effects that we're seeing in endothelial cells. Finally, we took this inhibitor cocktail in vivo, and what we did here in our local model of vascular leak is we injected mice intraperitoneally with the inhibitor cocktail. 6 hours later, we followed with another dose of cocktail along with our injections of PBS, VEGF, and the 2 concentrations of NS1 intradermally, then also the tracer of Alexa. And then finally, 2 hours later, tissues were collected and processed. And what we find here is that when we look at fold change from PBS, we see that we get induction of vascular leak with VEGF and NS1 as we saw before when these animals are untreated or are mock treated with a vehicle control. but when we treat the mice with the actual inhibitor cocktail, we see a decrease back to PBS levels of the NS1-induced vascular leak. So this inhibitor cocktail is reversing the vascular leak we observe in our local model. You'll also notice a slight decrease in the VEGF, and we think this is related to cathepsinel potentially playing a role in the VEGF pathway. And then also over here, the 7.5 micrograms of NS1 didn't turn out to be significant, but this was trending towards significance, and I do believe it would tighten up with a couple more animals. So, in summary, we found that dengue virus NS1 cytokine-independent vascular leak is dependent on some endothelial glycocalyx components. We showed that NS1 can directly trigger vascular leak in the dorsal dermis of wild-type mice in the absence of viral infection. We showed that human dermal endothelial cells do not produce the canonical pro-inflammatory cytokines TNF-alpha or IL-6 in response to dengue 2 NS1 stimulation, and also that these cytokines play no role in NS1-induced endothelial hyperpermeability as measured by TIR. We also showed that TLR4 and TNF-alpha signaling do not contribute to NS1-mediated vascular leak in our in vivo model. And finally, that we can inhibit endothelial sialidases, heparinase, and cathepsinel, and prevent induction of vascular leak by NS1, both in vitro and in vivo. Finally, I would like to thank all of the wonderful people who I work with in the Harris Lab, especially Eva, who's a fantastic mentor, and Kalani Ratnasiri, who is an undergrad who worked with me and did the majority of the mouse work shown in this presentation. She's a fantastic researcher, has a very bright future ahead of her. I'd also like to thank Henry Portaguardo and Diego Espinoza, 2 postdocs who did a lot of work with some of the systemic models in this talk, and also Dr. Robert Beatty, who helped design a lot of these experiments and interpret the results. And if you're interested in more of this work, this paper is going to be published within the next week or 2 in PLOS Pathogens, so keep an eye out for that. And with that, I thank you for your attention, and I'm happy to take any questions you may have.
Lovely work. I'm just trying to connect back to what it means to the natural history of clinical dengue in humans. And we know that some patients will develop dengue shock syndrome and NS1 antigenemic negative for the duration of their illness. We know that some patients with primary dengue 1 infections will go home having recovered and are still NS1 antigenemic. And I'm also trying to put this in the context of secondary dengue and enhanced risk. So lovely work in the skin of a mouse. I don't understand how it relates to clinical dengue.
For sure. Definitely get that question a lot. And we know that NS1 is a piece of the puzzle. Dengue pathogenesis is complex, and there are many things at play, potentially antibody enhancement. We are only touching the surface of what cytokines are doing here. There's a lot going on, and we definitely appreciate that fact as we go through this research in the Harris Lab. But we think this is a really interesting aspect of NS1 as a protein alone, the fact that the protein alone can induce these very interesting effects, both in vitro and in vivo model. And as you mentioned, this is in the back skin of a mouse, which is why we're trying to take this out to some more systemic models and look at the effects of NS1 on a more whole animal level. And we know that's not quite the same as looking in a human. But it does offer some potentially interesting or unique pathways to prevention and treatment of disease that we have to further investigate.
After the degradation of the glycocalyx, how long does it take for this to naturally regenerate? And are there things that will make that more rapid?
So in our in vitro models, we observe recovery of all of our glycocalyx markers by 24 hours post-treatment. Clinically, we know that dextran can help reverse some of the effects of vascular leak and has been implicated in helping to regenerate the glycocalyx. So it's a possibility that dextran itself may be a potential treatment for recovery of the glycocalyx. Thank you.
Thanks, Dustin, for the